At the synapse, where you get diffusion of neurotransmitters across the gaps between one neuron and the next. You might not have done that yet. You'll see also it happens in the leaf, it happens in the stomata, and so on. It's how gas exchange occurs in plants, is by diffusion. Okay. So this is a special case of diffusion. So osmosis is specifically to do with the diffusion of water molecules. Okay, so if it's the movement of water, then it might be osmosis. Not necessarily, but osmosis only involves the diffusion of water. So it's a special case in that sense, right, of diffusion. But it's also a special case in another sense. It's a special case because osmosis is the movement of water molecules from where there's a high concentration of water molecules to a low concentration of water molecules. In other words, down the usual concentration gradient. But it's weird this time because we're talking about the concentration of water. So if you look on the left-hand side of this, the cup on the left-hand side of your screen, there's, let me just make sure I have my chat box open here in case you want to throw a question in. And I also have in case there's anyone knocking on the door, I have to have all these little windows open here to make sure somebody's not outside the door trying to get in. Okay. So on the left-hand side of the cup on the left, the container on the left, you see the water molecules are in black. And there's way more water molecules on the left-hand side of the dotted line than there are on the right-hand side of the dotted line. So the left-hand side of the dotted line, that's a high concentration of water. It's a high water concentration. Okay. Now, it's weird because we look on that solution, that left-hand side solution, that's what we call a dilute solution, right? But it's a high concentration of water. That makes sense. On the right-hand side is what we call a concentrated solution. It's concentrated because it's got a high salt concentration, but actually it's got a low water concentration. So now the dotted line is the next thing we need to talk about with osmosis, because the dotted line represents what we call a semi-permeable membrane or a selectively permeable membrane. It's better to call it selectively permeable. What does semi-permeable even mean, really? Semi-permeable, and I use it as well. But if you think about it, half permeable, that doesn't really make any sense. It's stuff either gets through it or it doesn't get through it. Selectively permeable is a much better description for what we're talking about here. And we've already come across selectively permeable membranes because the cell membrane is one of those. So if you've done the experiment on osmosis, we use a model cell membrane. It's a model membrane, a model for a selectively permeable membrane. In fact, it is a selectively permeable membrane that we call Viscing tubing. That's what most people do. I don't know if you use Viscing tubing. If you haven't, then it's just it seems like it's like a cellophane or cling film or something sort of material. And it's kind of like a cylinder. It's in structure, and you can tie both ends and fill it with solutions and tie knots at the end and make it kind of like a sausage kind of a thing. So we'll look at that in a second. But so osmosis is two things. If you're going to describe osmosis, you need to make sure that you have two things in your description. It's a two-part definition. It's the movement of water, first of all, or the diffusion of water, but even just the movement of water from an area of high water concentration to an area of low concentration. So that's the first part. It's that it's about water. The second part is that it's across a selectively permeable membrane. That's really key. If both of those things aren't happening, then it's not osmosis, right? So for example, here you notice that what the salt does is of no interest here. What's happening is that the water molecules are diffusing through the selectively permeable membrane from the left-hand side to the right-hand side until the concentration of water molecules is equal. Not the number of water molecules, but the concentration of them, right? If you like, the density of them, right? The concentration of them is better. So, in other words, what happens is water molecules move through that, diluting the salt solution until the concentration of water in both of those sections is the same, and then osmosis will stop. Okay, so that's the definition. Really important: high water concentration to low water concentration across a selectively permeable membrane. So osmosis is that special case of diffusion. That was actually asked as a question one year. Why do we consider water to be a special case of diffusion? And what they were looking for in the answer was, just the definition of osmosis. If you gave them the definition, the full definition, then it's a special case because one, it only involves water, two, it's across a selectively permeable membrane. So like I said, we know about selectively permeable membranes because we've looked at those when we're talking about the cell membrane already. So this is the visking tubing I was talking about. It's this, yeah, it's kind of like a cellophane material. If you think of if you know the inner tube of a bicycle tire, it's like that, except made of cling film kind of material, or kind of a slightly more rigid than cling film, kind of clear plastic. And it seems like it should be waterproof or watertight, hold water, and it does, except that it's porous. So it allows, but it's based on size. So small molecules can get through, big ones can't. So sugar molecules are way too big, for example. So in this situation, we've got on the left-hand side, you've got your test setup, your experimental setup, which is visking tubing, which has got sucrose solution in it. And if you put that into distilled water, well, what happens is the water will diffuse into this visking tubing to try and, in order to dilute the sucrose solution, because there's a higher water concentration outside the tubing than there is inside. So water moves into it across the selectively permeable membrane that is the visking tubing. On the right-hand side, you've got a control. So what happens in this experiment, and we're going to look at experiments in detail later on in the year. But what happens is, after about half an hour, you notice that the visking tubing swells up, it becomes turgid. You take the mass of it, it's increased in mass. You look at to measure the volume of it, it's increased in volume. Why? Because of osmosis. If you take a red blood cell and you pop a red blood cell into pure water, it doesn't do well, right? It'll swell up because water will rush into it because of osmosis. It'll swell up, and because red blood cells, like lots of animal cells, don't have a cell wall to prevent them from bursting, it'll burst. So red blood cells don't do well. Red blood corpuscles, notice the word. Technically, they're not really cells, right? Because they don't have a nucleus. They don't have any mitochondria. There's not much going on in a red blood cell. It's just really just a kind of a little rubber dinghy for carrying oxygen around the place. That's its job. That's its only job. It does it really well. But they don't do well in pure water. So if you take a red blood cell, and you put it into a really strong salt solution, like a concentrated salt solution, then water is going to leave, because there's a higher water concentration now in that situation inside the red blood cell than there is outside the blood cell, where it's just loads of salt. So water rushes out by osmosis through the selectively permeable membrane, and the poor little red blood cell dribbles up and probably won't survive. So that's what happens. Don't worry about these crenation, hemolysis words. We don't need those words, but you just need to know the effect of if you put a red blood cell into either into pure water or into strong salt solution. Okay, if you put plant cells if plant cells are placed in water, and this is generally the state of a plant cell, right? Usually there's lots and lots of water inside the stem or the leaves or whatever of a plant. And we'll see that. We'll see why that is in a minute. So plant cells are highly adapted to be able to take on this loads of water. They have a huge central vacuole that swells full of water. Water enters by osmosis, and because the vacuole swells up, it causes the cell membrane to press against the cell wall. And you get this internal pressure, buildup of like water pressure, osmotic pressure, they call it, which causes the cell to be swollen, what we call turgid, right? So the cell wall stops the cell from bursting, like the poor red blood cell. And that means that now you have these plant cells that are, like I said, swollen, or the technical term is turgid. We need to know that word. Now, what's the big deal about that? Well, this is how plants stay upright, because at least soft stem plants are able to stay upright because of this turgidity of their cells. So the tissue becomes swollen. And if you notice, if you don't water a plant, I mean, lots of things go wrong if you don't water plants. But if you notice that if you don't water a plant, if you've ever gone away on holidays, you come back, and if you're in time, the plant will have wilted. In other words, it'll have fallen over because the turgidity of the cells has been lost, right? They're not swollen anymore. The plant can't stand upright anymore. So it literally just wilts and becomes limp. And if you catch it in time and you water it, within sort of half an hour or so, or an hour, the plant will stand upright again, because water will be able to take up the water by osmosis, and water will move from cell to cell by osmosis from the roots up to the stem and so on. And it has specialized transport systems as well to carry the water all around the plant. So we'll see how that works in a second. So turgor pressure. Turgor pressure is the force of the cell contents against the cell wall in plant cells. And it's, as I said, due to osmosis, right? Of course, if you put a cell into strong salt solution, do the opposite. In other words, now it's not full of water, it's going to lose water by osmosis for the same reason. And what happens is the cell membrane pulls back from the cell wall. And there's a gap then forms between the cell wall and the cell membrane. And those cells are what we call plasmalized. Okay, you know, if all the cells start to become plasmalized, then you get loss of pressure in the tissues, and then you end up with wilting plants, like we said. So that's the system there. You can see on the left-hand side is that plasmalized cell where the cell membrane has those pointers are in the wrong spots. Don't worry about that. So the cell membrane is the internal, it's pulled away. And then the cell wall is the outer layer. On the right-hand side, you can see that the cell membrane is pushed up against the cell wall because the vacuole is so swollen. Yeah. So we've got other examples. I just want to mention them. You may not have come across all of these yet. We haven't looked at amoeba. We still have to look at that. We put that on hold a few weeks back. The nephron, you may or may not have done already. That's in the kidney. That's an example of the reabsorption of water by osmosis, for example. And we'll see that the nephron features in active transport in a second. You've got plant roots. We talked about. We'll talk about roots root hairs rather. Are these, you know, what you might think of hair-like structures on the surface of the root to increase the surface area for absorbing not just water by osmosis, but other materials from the soil water as well, like minerals that they need. So that's another example of osmosis. And the stomata, if you know about the stomata, we'll look at that in a few weeks' time. The guard cells, how they open and close, we'll look at. But the basic idea is that when they open, then, why they open is to do with the intake of water by osmosis. So those first two are examples of what we call osmoregulation. So osmosis is really important process in not just in single-celled organisms, but in multi-celled organisms, because all organisms are constantly trying to maintain their internal environment, right? The concentration, the amount of water, the concentration of the dissolved solutes, all of the temperature, all these things. But when we're talking about just the water levels and the dissolved substances in the water, in cells and in tissues, in blood and in other body fluids, that has to be maintained. For example, in us, like in multicellular organisms, has to be maintained at a very kind of steady state. So the functioning of all of our metabolism, our enzymes and all those things relies on us being able to maintain this internal environment, which we call homeostasis, right? But when we're talking about just looking at the amount of the water and salt levels in our body, we talk about that as being an example of osmoregulation. So you can see osmosis and osmoregulation are obviously related words, right? So it's to do with the water levels, the water concentration, and therefore also the salt concentration or the dissolved substances concentration, so sugars and things as well. Of the internal environment, we call that osmoregulation. So this is the definition: maintenance of the internal balance between water and dissolved materials. Just, you know, regardless of whatever's happening in terms of the environmental conditions. So, for example, if you're swimming in the sea, that shouldn't, your body is able to ensure that the internal water and salt levels are unaffected by the fact that you're, immersed in salty water. Okay. So how it's done largely in humans is by the kidneys, and we'll come to that another time. In microorganisms, we exploit this, the plural microorganisms. So we know about osmosis, and so we can use that against these microorganisms who are trying to grow in our food, for example, right? So let me just, yeah. Okay, super. So, for example, if a fungal spore or a bacterial cell lands on, let's say, jam to which we've added loads of sugar, or some salty meat that we've added loads of salt to. So what happens in either of those situations is water will flood out of the bacterial cell or the fungal cell by osmosis, right? Because there's much more water inside the cell than there is in the very sugary or salty food that it's just landed on. So water will leave by osmosis and it'll shrivel up and die. In that way, we can preserve food by either salting it or adding loads of sugar. So hams and jams, right? And so bacteria in general, microorganisms don't do well. And therefore, that's a good way of us preserving our food. It doesn't always work. The sugar level has to be high enough for microorganisms to just not be able to survive and for the osmosis thing to take care of them. But of course, some microorganisms are highly adapted to live on very salty or very sugary foods. So especially low sugar jams should really keep them in the fridge, or eventually some mold is going to be able to grow on it.